And we'll welcome Sergi Castellano to the stage to chair the next section.
It's truly a pleasure to introduce the genomics theme. Just word of advice, unfortunately, Dr. Laurent Daldi cannot be with us today due to personal circumstances, but we will still have two brilliant speakers sharing talk. But we first have a video on the theme. So the overall goal of the genomics theme in the GOSH BRC is to ensure that we move from gene identification to treatment to reduce burden of disease in children. And to do that, we apply a wide range of techniques. So we collaborate with patients and the public early to develop our grants, and that allow us to make sure that the results of the research benefits them. So we leverage extensive expertise at the Great Ormond Street Hospital and University College London. And for example, we have already contributed to some large consortiums like the 100,000 Genomes Project, COVID sequencing, and many of these results in genomic expertise is later translated into the NHS, for example, at the North Thames Genome Laboratory Hub.
A major success of the theme so far has been the development of rapid sequencing and cell-free DNA analysis. This has led to patients-acceptable, more rapid, prenatal non-invasive diagnosis, which has now been incorporated into the National Genomic Test Directory. This has also enabled us to help thousands of families and to deliver diagnoses more rapidly to these families. We cover a wide range of topics in the genomics theme, including improving diagnosis via whole genome sequencing and finding new genomic links to disease. We also are doing this via improving the cutting-edge pipeline which is existing within the theme, and also better understanding the links between infection and genomics. This is done in a variety of locations throughout the theme, including co-location of theme scientists within the local NHS Genomic Laboratory Hub. So a major area of growth will be working with the newest theme in our BRC, Applied Child Health Informatics. This will enable us to gather large amounts of clinical and genomic data in order to define new diseases, to undertake risk prediction, and to develop novel personalised therapies.
Hi, my name is Laurent Daldi. I'm the Head of Research and Innovation Communications at Great Ormond Street Hospital, and my talk was entitled Innovation in Communications for the BRC showcase here at Great Ormond Street Hospital. But when I sent over my title, I was a little concerned that the audience might think that I was going to be talking about AI or robots, so I wanted to manage their expectations by giving it a subtitle of Windows, Walls and Working Together. And it's really the privilege of me and my team to tell stories of research and innovation from the hospital and our partners. And this talk is really about how we use some of the principles of research and innovation in our work in communications. So whether that's telling stories in a new way or finding answers to old problems, and how we can apply that to communication. So what I'm going to cover is two old questions, so existing questions, two ways that we answered them and some impact of that. And then I'm going to wrap up with some plans for the future. So the first big question I wanted to answer was, how do we tell the whole family of stories? And that might sound like quite a broad question, but hopefully, depending on your term of reference, you'll recognise some of these very famous families. And I'm sure you can all agree that if you only told the story of one member of any one of these families, you'd be missing out on a lot of other opportunities. And they're really the same as in research. So we tell research and innovation stories often in the same way. So it might be a world-leading professor leads a cure, or NHS saves lives for children, or the first patient in a clinical trial. And this is great, and this is always going to happen this way. These are the huge game-changing breakthroughs that we love to see in the press. But like with those families, there's only one member of the family story being told here. And there are thousands of research firsts and innovation moments, and what I like to call the sort of well, I never moments, it could be just as interesting and inspiring. And if you only tell stories in this one way, we're never going to see that part of the family. So what we did in the Research Innovation Communications team is we launched a competition. So with funding by our Biomedical Research Centre, we launched that competition 2 years ago, and we really focused on the impact of the image that was derived from research or innovation, and then we mined those images for their press appeal. So we've done that now for 2 years. We've had 2 competitions. We've had things like live voting at a BRC event. We've had some social media voting. We've had the usual things like panels, and we've now had 6 panel favourites and 2 overall winners. And that's been great, getting that process up and off the ground. But that's actually led to 2 BBC stories, 1 in Wired magazine, 2 inclusions in Nature Images of the Month, and 1 in Nature Images of the Year. And yes, that is Nature. 2 temporary exhibitions at the hospital using some windows. So there's my windows that you can see there, beautiful displays of the images. And they're actually going to be worked into some permanent displays in the trust soon, we hope. And those images are also being used throughout the hospital in our materials. So anybody who's publishing a strategy or looking for a button for a website is almost always using these images now. And we've even been approached by our peers for advice on how to launch their own competition. So that's what we did. But the outcomes of those things that we did in terms of telling stories from the whole family have really been great. So we've worked with over 25 researchers and innovators that have been shortlisted, from PhD students to professors, nurses to physicists. So there's been quite a broad range of staff roles and job types represented. 10 disease areas, maybe even slightly more, with microscopy all the way through to illustration in terms of image type. And what I really like about this project is it's given us the opportunity to tell stories of research that's not really finished or hasn't had results yet. So it could be newly funded grants or something that's not quite publishable, collaborations, or even some images people didn't think were good enough for publication have made it into this competition and have been showcased in a new way. It's given us a chance to work with our young people's panel through YPAG, and also to have a diverse shortlisting panel in the second year. And this is all about building our new networks for research and innovation champions. I don't think this is the answer to the story. I don't think it's going to fix that question overnight, but it's the start, and it's the start of building up those networks. So our second big question is, how do we make the hospital look and feel like an intelligent research hospital? Now, another big one, and I don't think we're going to solve this overnight, but if you have a little look around online, you can see some pretty dramatic images of what a research and innovation hospital might look like. Perhaps not totally realistic, and you'd be glad if you were Judy Jackson, I think, having this procedure. But there definitely are places in our hospital that do feel and look that they have research and innovation happening, and that's right. But the way that our hospital works is that research and innovation are embedded in the very fabric. Research is not just in the labs, it can be on the wards and the clinics, and you can't always see it, and the same is true for innovation. So how do we bring this out in a meaningful way for our staff and our patients? Now, as a communications team, we got our heads together and we thought of all the incredibly striking things we'd seen that raised awareness for us in the last sort of 5 years. And we came up with a real broad strokes of different things. And we got really carried away, and then we realised we had to stop. And the aim of what we were trying to do was to share a message of an intelligent research hospital. And hospital was that operative word. It couldn't really get in the way of what we wanted to do, what we essentially want to do as a hospital. So we realised that our 3 key objectives, after discussing it with patients and staff, should really be raising awareness of research and innovation, doing something that allowed people to see themselves in research and innovation, and they should also be engaging but not overwhelming. So I don't know if any of you have tried to get a child to an appointment on time for absolutely anything, but usually it's pretty good luck if everyone's got all their shoes and socks on. So we didn't really want to create anything that would be a barrier to getting often small children to a place on time. And many of our patients have learning needs or sensory needs that we needed to make sure we had taken into account too. So the first thing we did was we found some unused spaces in the hospital, and this is where my walls come in. And we had some existing illustrations as well as some permissions to use both. We created some designs that reflected how staff and patients are really integral to research and innovation. And then we took the opportunity to invest in really dialing up the diversity of those illustrations. So we paid an illustrator a little bit more money to develop the character. So what we have now is a lovely lift, as well as the doors, that really show patients how they can be integral to research and innovation, but also directs them to clinical staff, not a separate research team, if they want to get involved or have more information. We have a staff wall in the back of the lagoon, our staff canteen, that signposts some vital research infrastructure and our innovation hub. We had an all-staff launch, which was great, thanks to our chief exec, who launched that for all staff online through our all-staff messaging. And we've also put it on social media. So even if you can't get into the hospital, you can get involved in the messages these are sharing. And it was a really lovely way to highlight research and innovation, messaging, how you can get involved, and all the very, very different kinds of staff and patients involved in that. So again, these are things we've done, but what has that actually meant? Well, quite quickly, even before we'd finished, we had much better patient and family involvement from the team. We developed these in collaboration with people in and out of the hospital every day. We've had lovely positive feedback from patients, staff fora, and various research stakeholders right from the get-go. And the social media posts that we've had about this had the highest engagement rate for the month. So it was really, really warmly received. It was really a chance to show a good breadth of diversity of staff and patients. I don't think it's perfect. I think there's always more we can do, but we'd already moved on from the illustrations we developed 2 years ago to realise how much further we could push them. And what was really, really gratifying to see was some lovely feedback we had from our co-chair of the REACH network, which is a staff, one of the staff diversity forums at Ormond Street Hospital, essentially telling us that what we'd done was really good and they'd love to see more of it. So we took that as a really good ringing endorsement. So again, not fixed, not answered the question entirely, but we're definitely on our way. So now, of course, is how we build on this for the future. Well, it brings me to my final W, which is working together. And I really do believe this is going to be an area we can improve upon. We do some of it already. So in terms of working with our partners, be that funders or collaborators, local or global, we do some communications with them, but we could always do more. It's working with our patients and young people. We have teams that do this at the hospital and do this really well within research, but how can we lead on the comms to do that? So could that be taking an event where Steve Khan plays with water on the hottest day of the year with lab coats for the children to get us in some media exposure, or relying on a man in a bear suit wearing a lab coat cuddling a dog, or all open for discussion. And then we've got our networks, so our champions network for staff, and we've got research badges and certificates, as well as for the children to bring people together. And then on a smaller level, we've also got our internship. So last year saw our first research communications internship, and if you've seen any of the illustrations from the BRC showcase and the ones you can see here, they were the results of a very talented postdoc who came to work with us for 3 months. So essentially, it's all about making sure that our internal messages, the messages we're telling our staff and our stakeholders, matches the external messages we're sharing so that everybody can see their place in the stories. No research talk would really be complete without a long list of thank yous. So thank you for listening, and if you've checked into the video. But the Research, Innovation, Communications and Engagement team is really a rather long name for 3 people, myself, Fiona and Gigi. And Fiona in particular has led a lot of the work that you've seen today. So big thanks to them. We're funded by the BRC, the Research and Innovation Directorate and Gosh Drive, but we really are supported by a whole breadth of teams at the hospital, external communications, external, all the way through to Gosh Arts and the Learning Disability team. All our researchers very kindly take the time to share their stories with us. Our innovators give us all their great ideas so we can share them widely. But it's our families and carers, I think, that always go into making this possible, so we can bring the stories to life and have the privilege of working with them to share their stories. Thank you very much.
And now it's a pleasure to introduce 2 speakers. Dr. Natalie Chandler, who is the Senior Clinical Scientist in the Genomics in the North Thames GLH. So she's at the forefront of implementing some of these technologies in the clinical setting. And she will discuss some of something that Thomas has referred to already, which is prenatal diagnosis. And then we will have Dr. Amy McNack, which is an honorary consultant on pediatric neurology, and she will discuss some of her work using genomic sequencing on children's epilepsy that she does here at GOS ICH, but also in large consortiums with some of the best pediatric hospitals in the world. So, Natalie.
Thank you. So I'm going to talk about the development of rapid sequencing for prenatal diagnosis. So when we first started this, the standard of care firstly was QF-PCR to look for the common aneuploidies, which explained about 30% of scanning anomalies. And then there was karyotyping available, which explained another 5%. And then there was RACGH, which then explained another 6%, leaving around 40% undiagnosed. Sorry, 60% undiagnosed. Of those, it was estimated around 30% would have a single gene disorder, which in the case there was very limited testing back in about 2017, 2018. So there was this study, the page shows the prenatal assessment of genomes and exomes. So this was started around 2012, and this was to look at the clinical utility of doing sequencing in a prenatal scenario. So this was done in a non-urgent manner. It was just to evaluate it, so all results were returned post-pregnancy. We accepted any fetal anomaly, and there was no review of any of the findings. So the outcomes of the study showed there was clinical utility to doing sequencing, but it depended on the different type of anomaly. So skeletal phenotypes, there was about 20%. However, if you went down to just large nucleus, etc., it was only 4%. So following that, because we've shown within the page study that there was clinical utility doing the sequencing, we wanted to develop it within a lab and set up rapid testing pathways and look to see if it was feasible to do this testing in a rapid turnaround time to ensure that it can have an impact on pregnancy management. So within the BRC research team, we did this study on skeletal dysplasias, having shown to have the highest diagnostic yield. So we used exome sequencing and we applied a panel. And what we did for this study, rather than the page, is we looked at every single referral to decide whether it was likely to be genetic or not. So therefore we gatekeeped all the referrals, which meant we then ended up with a very high diagnostic yield of 84%. We were able to test all samples where they'd had invasive testing, so we had enough DNA, and we were able to do the testing in an average turnaround time of 10 days, showing that it was feasible for us to do this in service. What this also allowed us to do was to train up our scientists, develop our pipelines, and train our lab technologists, and get all the validation data so that we were ready to implement this into clinical service, and also present the data to NHS England to fund. So following these studies, the NHS agreed to fund this into service, which started on the 1st of October in 2020. It was decided we would offer the service for half of England, along with the Birmingham laboratory who do the other half. So we have the outcomes from the first year of service. So the diagnostic yield matches similar to the yield for exome sequencing postnatally, so around 30 to 40%. The first year we had a yield of 40%. We were able to deliver in a mean turnaround time of 14.6 days. In the audit data, we've also looked at the evaluation of the service. So it's been shown that the exome sequencing result has impacted the management of the pregnancy in 56% of cases, 79% of those that have got a diagnosis, and for 37% where we didn't find a diagnosis with the testing. So these are three just examples of where they've impacted. So first is a diagnosis of a primary ciliary dyskinesia, and getting the diagnosis in this case meant that there's early respiratory team involvement, and we were also able to test the siblings. Another case, we got a diagnosis of Silver-Russell syndrome in a case of IUGR. This meant that normally you would intervene in a case that had growth problems and deliver them earlier, but because there was a genetic cause, they allowed them to continue to birth rather than early intervention. Another case of Holt-Oram syndrome, because this is seen as a kind of more treatable condition but they have normal intellect, etc., and they can have surgery to treat the conditions. Getting that diagnosis gave parents confidence to continue their pregnancy. So what do parents and professionals think? They're really pleased with the speed of the results. They think the communication and working with the MDTs has been really good and really been appreciated. So fetal medicine, I think it wasn't working well at the start. It was quite a complex test to set up, but we've worked strongly with the clinical genetics and the fetal medicine consultants to get a more streamlined process going. It's now working better. And from patients, so it would have been helpful to have a conversation with someone after they took the sample just to really go over things. So there's still things we need to address, and it's still very challenging in an urgent environment to be able to give the right counselling to patients. So there's still things we need to work on. So we're not done yet. So this is all done on whole exome sequencing, but ideally we want to move to whole genome sequencing. That is what we're currently working on. Why do we want to move to whole genome sequencing? But it actually to the whole genome, whereas the exome, we've just got the gaps. We've got gaps, so we're only doing the coding region of protein coding genes. WGS gives more uniform coverage and complete coverage of genes, and there's very few errors compared to exome. It's much quicker library prep, and you can therefore do things in quicker turnaround times. It's much better for calling chromosomal abnormalities, and hopefully it means we'll be able to stop having to do microarray testing. So it's already on offer in the NHS for routine testing, but very slow. So the hope is to bring it into our lab and be able to do it in a rapid format, similar to what we provide for exome sequencing. And being able to do WGS means that we won't have to resequence after birth if they've got new features, because at the moment, if they go into the routine NHS, they'll then have to be resequenced. We do have a lot of challenges to address to bring this into service, so data storage, DNA input, developing our bioinformatics pipeline, we need to do validation, and also how do we deal with the increased costs. So that's why we're doing pilot studies on rapid WGS before we can clinically implement it, just like we did for exome. So the prenatal scenario is one of the indications we're looking at, but we're also looking at other indications. As to Amy.
Thank you very much, Natalie. So the early onset epilepsy has affected about 1 in 1,000 children in the UK under 12 months of age. And about 50% of these patients will have a genetic cause that can be identified after serial genetic tests. And unfortunately, many of these babies have severe epilepsy known as developmental and epileptic encephalopathies, where they have frequent seizures, very abnormal EEGs, and they tend to have a poor developmental outcome. And we now know that over childhood, about 1 in 600 children will develop a DEE. So although these are individually rare diseases, they are collectively not so rare. In addition, we know that epilepsy is genetically rich in terms of gene discovery, as we've seen over the last 20 years. And there are indeed some advantages of having a rapid or a timely genetic diagnosis. So for the most commonly identified genetic things that cause epilepsy, if you have a loss of function SCN1A mutation, we would never use sodium channel blockers, but for children with gain of function neonatal seizures with an SCN2A mutation, we would use sodium channel blockers as our treatment of choice. So having that early diagnosis allows us potentially to select the right treatment to avoid things that make things worse and to maybe start to implement precision medicine for these children. More long-term, it allows us to do accurate genetic counselling to avoid unnecessary tests, of which these babies usually have many tests. And also, we think importantly, to identify early developmental and cognitive vulnerability. So I was very fortunate when this consortium started. This is the International Paediatric Child Health Precision Medicine Consortium, and this was started between GOSH and several of the world-leading children's hospitals in order to really harness the promise of genomic medicine for precision medicine for children. At the time, I was a Catalyst Fellow, so I've been lucky enough to receive one of the BRC Catalyst Fellowships, and this allowed me to get a further fellowship, a clinician scientist fellowship. And it really was a major part of my development as a researcher to be able to lead the infantile epilepsy group across this consortium with the support of my senior colleagues, Professor Cross and Professor Chitty and others. And this allowed us to devise this study, so gene steps. So we're essentially saying that we think for babies who have early seizure onset, rapid genetic diagnosis will reduce these unnecessary investigations, allow us to choose the right treatment, and potentially improve the developmental and cognitive outcomes. And also, most importantly, we want to look at what impact that has for patients and carers. So again, we were fortunate to benefit from a new project award in 2021, and with further funding from other partners, including Gosh Charity, we're able to launch our pilot study in December 2021. So what this essentially means is that we undertake recruitment with rapid trio whole genome sequencing in the proband and parents. We undertake deep phenotyping and developmental assessments at different time points. And we're doing really detailed prospective developmental assessments, as well as looking at parental stress. We also plan in the next phase to do qualitative research with the social scientists to look at the impact for parents. And this has really been possible because of the unique placement of the BRC between ICH and Gosh, and our ability to look at the unique patient population that we serve. In addition, alongside with the Child Health Informatics theme, we've been able to look at developing new informatic approaches, including a red cat tool, which is being used across the 4 centres across very different healthcare settings. So this is our flow. So we identify the patients. We just saw a patient this morning who's going to be sequenced hopefully in the next few days. And we then use the NovaSeq in Gosh to sequence these samples, and we return samples aiming for less than 21 days, which is obviously much faster than the current NHS standard of care. This is an illustration of our turnaround time. You can see we've got a median of 17 days. We do have a few outliers which have largely been around patients who've had initial diagnoses that have been difficult to resolve, or where there have been complex kind of microdeletions or duplications. Overall, we've got about a 50% pickup rate, which I think you'll agree is fairly high for a diagnostic test. And most importantly, or as importantly, across the international cohort, we've also been able to deliver this result before 21 days for most patients. And also, although these patients take different amount of time to present to services, we also get a diagnosis back quite quickly after seizure onset. Overall, our pickup rate across the 4 centres internationally is also about 43%. This is much higher in the neonatal group, which has been shown before, but also varies by syndrome, by whether there's microcephaly and whether there's pre-existing developmental delay. So our challenges and next steps include that we want this to be available to not just patients at Gosh, but more broadly. So our initial next steps are to roll out across the genomic hub territory, across North Thames, but we'd like to roll this across more widely because we think we're seeing real clinical utility and impact of this rapid diagnosis. We're advocating with our partners in the BPEG and the BQNA for equity of access. We're also undertaking functional evaluation of some novel genes, and again, we're fortunate to receive some funding to undertake some long-read sequencing in collaboration with Chandler and Chitty on some of the unsolved patients. And lastly, we want to harness the data that we have from these patients, and we're looking at working across international borders to share data in conjunction with colleagues such as GA4GH, International Genomic Data Sharing Authorities. So we'd like to finish by thanking everyone that we work with, particularly our colleagues in the hospital, within the BRC, and also with all the clinicians we work with, but particularly thanking the patients and their families. Thank you very much.
Thank you very much, both the speakers. Questions?
Hi, there's a couple of questions on Slido. So the first question to either of you, it says the advantages of rapid genome sequencing are clear for everybody. Do you think we'll eventually reach a situation where everybody is sequenced at birth rather than needing to move?
I think the issue needs to be addressed there is data storage and the cost involved in that. So I think that it's astronomically expensive to save data and then reanalyze it. So I think unless that comes down significantly, we will not be doing it.
Really beautiful talks, thank you. For some of our research studies, we're also doing whole genome, and we've come across this issue about how to counsel on other variants that are nothing to do with the clinical phenotype, but may be important later in life. So 2 questions related to that. Do you have a standard policy, and is there a wider need for training, perhaps at medical student level, for really understanding how to explain this kind of data?
Yeah, we've already had 2. So it's definitely an issue. So because we're doing an agnostic analysis, not a panel-based analysis as is done currently in the NHS, we do, when we do our consent, we do counsel parents that we have an increased likelihood of identifying an additional secondary finding. And we say that if this impacted on the health of the child, then we would take this to an MDT with our genetics colleagues, and we would discuss the appropriateness of divulging that. We have had a couple of situations where that has been inherited from a parent, and we've then had to counsel parents as well. But we're very upfront consenting. We make sure that we really outline that clearly, because that's quite different to the NHS sequencing, though similar to some of the 100,000 genomes participants who opted into those findings. It's been interesting doing this internationally, because it's a very different approach in North America and Canada. Australia was similar to us. In Canada, they black box the adult onset secondary findings and don't look at them, but they do report the paediatric onset secondary findings for the child. And in America, they return everything. So I think there are conversations going on. It'll be interesting to wait for the outcome of the research from 100,000 genomes, where they studied the kind of what the impact of feeding back those results were. But yeah, it's an issue for agnostic analysis, certainly.
There's one further question on Slido, which says, at the moment, does everybody have equal access to whole genome sequencing if they're sick, so across the UK, or is it only here at GOSH?
So there is a national genomic medicine service, and everyone does have equal access to the kind of standard of care NHS testing. And there is access to emergency testing for patients who are very unwell in intensive care. What our study is doing, and this is only currently available to patients within GOSH, but we really want to expand this, and we're discussing with people about that. We're testing both kind of inpatients and outpatients, and patient coming to the day case unit. And in fact, in our big international study, there's the number of patients who are from those settings are higher than the intensive care settings, which is what all the previous studies have been. So we're trying to show that you need it outside those settings too. Thank you.
Thank you again. Can I ask Professor Fury? Your turn.